What's up mga kapakpak? Magandang morning sa ating lahat. Ako nga pala si Sean May Balanza aka Liza Sobera Noo. Before we start, shoutout ko muna ang aking mga ka-groupmates. Si Rica Turato aka Nadengue Lustre, Roxanne Tumitit aka Solane Use of Lungs, Justin Aranya aka Catherine Bernard Tugol, Juliana Clarinal aka Are You Lost Baby Girl, Angel Carino aka Lucy Fur, and last but not the least, Jasmine Talavera aka Aloe Vera. So let's now first tackle what is pyruvate. So pyruvate is a key molecule in metabolism of eukaryotic and human, and its fate differs depending upon presence and absence of oxygen. It is also the end product of glycolysis and is eventually transported into mitochondria as a major energy and participates in the TCA cycle. In the glycolysis, glucose is converted into two molecules of pyruvate with the generation of ATP. However, if reaction stops at pyruvate due to imbalance redox, it would not proceed for long. The enzymatic activity of glyceraldehyde 3-phosphate dehydrogenase produces a molecule containing high phosphoryl transfer potential and reduces NAD to NADH. However, NAD molecule is present in very limited amount in the cell and it must be regenerated for glycolysis to proceed. This is achieved by the metabolism of pyruvate. Pyruvate are mainly converted into ethanol, lactic acid, or carbon dioxide, which will be discussed by Justin Aranya.
First, I will discuss about the fate of pyruvate in the presence of aerobic condition. In the presence of oxygen, molecules like glucose and other sugars, fatty acids, and most amino acids are eventually oxidized to carbon dioxide and water via the TCA cycle and the respiratory chain. The carbon skeletons of sugars and fatty acids are converted into the acetyl group of acetyl-CoA and enters into the TCA cycle, the form in which the cycle accepts most of its fuel input. In the matrix of mitochondria, first pyruvate is converted to acetyl-CoA by the enzyme pyruvate dehydrogenase complex or PDC because farmers cannot enter the TCA cycle.
Breakdown of pyruvate. Each pyruvate molecule loses a carboxylic group in the form of carbon dioxide. The remaining two carbons are then transferred to the enzymes coenzymes A to produce acetyl coenzymes A. A carboxyl group is removed from pyruvate, releasing a molecule of carbon dioxide into the surrounding region. Note, carbon dioxide is one carbon attached to two oxygen atoms and is one of the major end products of cellular respiration. The result of this step is a 2-carbon hydroxyl group 1 to the enzyme pyruvate dehydrogenase. The lost carbon dioxide is the first of the 6 carbons from the original glucose molecule to be removed. This step proceeds twice for every molecule of glucose metabolized. Remember, there are 2 pyruvate molecules produced at the end of the glycolysis. Thus, 2 of the 6 carbons will have been removed at the both or at the end of the both of this step.
PDC is very important for health point of view as well. It is involved within degenerative neurological diseases, obesity, type 2 diabetes, and other diseases. More recently, PDC gained attention in cancer biology, which is mainly attributed to prominent role played by aerobic glycolysis in some cancers. Pyruvate can be converted to oxaloacetate in a reaction catalyzed by the biotin-dependent enzyme pyruvate carboxylase, and later molecule enter into TCA cycle to generate energy. It is an important step to replace the intermediates of the TCA cycle and make available as substrates for glyconeogenesis. It also involved information of aspartate via transamination reaction.
Let's proceed to the pyruvate converted to oxaloacetate. In the anaerobic state of pyruvate, it goes through fermentation that leads to the arrangement of lactic acid or alcohol. The response of the fermentation reduced NAD+ is generated, which indirectly help in synthesis of ATP in the glycolysis process. During the process of evolution, the earliest cells lived in the strict anaerobic condition and had utilized glycolysis as one of the approaches of metabolism and to produce NAD+ and as lactate or ethanol as final products. In the fermentation of lactic acid, its fermentation is the most common one. It happens in numerous microbes leading to form lactate from pyruvate. This transformation is likewise found in cells of higher animals under specific conditions like extensive exercise that would make oxygen-limiting conditions for muscle cells and would go through anaerobic respiration. So lactic fermentation is going inside our muscle cells when you only really do a lot of physical works. Because the glucose are burning out, then it starts to accumulate the lactic acid into your muscles, and you will start to feel the pain into your muscle. So in this process, the lactate is shaped from pyruvate in the response catalyzed by lactate dehydrogenase, and this is a significant advance to restore the supply of NAD+ so as to guarantee that glycolysis is long-lasting. In any case, lactic acid is toxic to cells since it makes an adjustment in the pH and leads to acidosis.
The pyruvate produced in red blood cells or the RBC by the enzyme lactate dehydrogenase is converted to lactate and is a lightly reversible reaction. So in this method, NADH is oxidized to NAD positive, which helps to reduce lactate pyruvate. So the pyruvate is converted into lactate due to a lack of mitochondria in RBC, the site of the TCA cycle. This step also helps to regenerate NAD positive and will further enter the site of ATP synthesis for RBC in the glycolytic pathway. So the lactate is also produced in muscles under vigorous muscle contraction due to exercise activities. So this leads to building up of lactic acid in the muscles, causing cramps, and it also causes pain.
In another process, under anaerobic condition, pyruvate is further metabolized to ethanol with regeneration of NAD and formation in a simple eukaryotic cell like yeast. Pyruvate is converted to ethanol with the liberation of carbon dioxide in a two-step fermentation process. These steps are irreversible reactions were catalyzed by pyruvate decarboxylase and alcohol dehydrogenase. TPP is a cofactor for both of these enzymes. In a first step is catalyzed pyruvate decarboxylase, in which pyruvate is decarboxylated and converted to acetaldehyde. The product acetaldehyde formed in this reaction serves as the substrate for the next enzyme in the pathway.
Alcohol dehydrogenase catalyzes the conversion of acetaldehyde in the presence of NADH to NAD, ethanol, and carbon dioxide. It is an important enzyme found in many organisms that including in humans. In the latest liver, it carries out the oxygenation of ethanol, which is either ingested or produced by intestinal microorganisms, with the concomitant reduction of NAD to NADH. Lactate and ethanol produced by microbial fermentation are commercially exploited for human use.
Chicken wing, hot dog and bologna, chicken and macaroni, chillin' with my homies. Chicken wing, hot dog and bologna, chicken and macaroni, chillin' with my homies. Hey, hey.